Today we are going to introduce enzymes in biofuels. First, we are going to introduce what biofuels are. Biofuels are solid, liquid, or gaseous fuels derived from the conversion of biomass. Biomass is organic material from plants, animals, or microorganisms, including agricultural crops, forestry waste, and municipal waste. Biofuels are divided into three categories: first-generation biofuels, second-generation biofuels, and third-generation biofuels. First-generation biofuels are biofuels produced from food crops, oil crops, and sugar crops through traditional processing, such as bioethanol, made from corn, sugarcane, or other sugary crops. It can be blended with petrol or used in specialized ethanol vehicles. Second-generation biofuels are biofuels produced from non-food biomass through biotechnological and chemical processes, such as lignocellulosic ethanol, biomass oil, and algal fuels, like biodiesel, made from algae or waste cooking oil. It can be used in diesel engines without any modification. Third-generation biofuels are produced from microorganisms by biotechnology, such as biohydrogen and biohydrocarbons. Third-generation biofuels are still in the early stages of development. They are made from new technologies such as algae or synthetic biology. Depending on the type of fuel generated, can be used in different places. For example, in transport, biodiesel can be used directly in diesel engines or blended with conventional diesel. Bioethanol can be used directly in petrol engines or blended with conventional petrol. Bioaviation fuels can be used in aircraft to reduce carbon emissions from air travel. In construction, biomass such as wood chips and crop residues can be burned directly for heating. Biodiesel, bioethanol, etc. can be used in boilers or heat pumps for heating. Next, in the field of power generation, biomass such as wood chips, crop residues, etc. can be used in biomass power plants to generate electricity, such as biodiesel, bioethanol, etc. can be used in gas turbines or boilers to generate electricity. Traditional fuels such as carbon, oil, etc. These fuels are non-renewable resources that will eventually run out. Secondly, their combustion produces large amounts of greenhouse gases and pollutants, leading to environmental pollution and climate change. However, biofuels are renewable resources and can be used as an alternative to fossil fuels to reduce the energy crisis. Secondly, biofuels emit less pollutants when burned and can reduce environmental pollution. Finally, biofuels can promote agricultural development as well as improve economic efficiency.
Enzymes are like nature's microscopic tools. They speed up chemical reactions, and they're the key to unlocking biofuel power. Much of biomass is tough. Think of wood, corn stalks. It's full of cellulose and starch. Enzymes break these complex materials down into simple sugars. Once we have those sugars, we ferment them to produce ethanol or other biofuels. Cellulases, hemicellulases, amylases, and specific enzymes are key categories in biofuel production. Cellulases, such as endoglucanases, exoglucanases, and beta-glucosidases, break down cellulose into fermentable sugars. Hemicellulases, including xylenases, mannanases, and accessory enzymes, target complex hemicellulose polymers. Amylases like alpha amylase and glucoamylase play a role in starch-based biofuel production. Examples of currently used enzymes include Celix-Tec and Accelerase. Cost-effectiveness and substrate specificity influence enzyme adoption, with advances in biotechnology driving the discovery of novel enzymes. Candida antarctically Passi B. Calb is highly specific for the transesterification reaction needed for biodiesel production. It exhibits excellent stability in the presence of alcohol, a crucial factor in the biodiesel process. It displays high activity even at moderate temperatures, optimizing the reaction. Calb is widely produced, making it readily accessible for industrial applications. Then is the Lipasi from Thermomyces lanuginensis. This Lipasi is known for its ability to function at high temperatures, potentially allowing for faster reaction rates and improved process robustness. It also shows good stability in the presence of alcohols. Used in biodiesel production, it demonstrates the ability to work with various oil or fat feedstocks, broadening its utility. In the context of biodiesel production, immobilization is a technique of attaching enzymes to solid support materials, resulting in enhanced enzyme stability, recycling capabilities, and improved cost efficiency. Immobilized enzymes exhibit higher resistance to harsh conditions, facilitating their use in biodiesel production processes. The ability to recycle immobilized enzymes minimizes waste generation and reduces production costs. Additionally, the extended lifespan of immobilized enzymes leads to reduced downtime and uninterrupted production, maximizing operational efficiency. Immobilization methods for Lipasi enzymes include adsorption, covalent binding, entrapment, and cross-linking. Adsorption involves the weak attachment of the Lipasi to a solid carrier, while covalent binding creates stronger chemical bonds between the enzyme and the support. Entrapment traps the Lipasi within a porous matrix, and cross-linking chemically links enzymes together to form larger, more stable aggregates.
What are the challenges of enzymes in biofuels? Firstly is cost. Enzymes can be expensive to produce in large quantities, making biofuel production less competitive with fossil fuels. Research focuses on developing cost-effective enzyme production methods and using them more efficiently. Second is substrate specificity. Most enzymes work best on specific substrates. Developing enzymes that can handle a wider variety of feedstocks, including those with complex compositions, would improve biofuel flexibility. Third is enzyme stability. Enzymes can be deactivated by factors like high temperatures, pH changes, and the presence of inhibitors in the feedstock. Scientists are engineering more robust enzymes to withstand these harsh conditions. What are the futures of enzymes in biofuels? First is enzyme immobilization and process integration. Immobilizing enzymes onto solid supports or integrating them into continuous production systems can improve their stability, reusability, and compatibility with industrial-scale biofuel production processes. Advances in enzyme immobilization technologies and process integration strategies will further enhance the feasibility and economics of enzyme-based biofuel production. Second is genetic engineering of feedstock. Genetic engineering of feedstock plants or microorganisms can enhance their suitability for biofuel production. This includes modifying biomass composition to increase its digestibility or engineering microorganisms for direct biofuel synthesis, potentially reducing the need for enzymatic hydrolysis steps. Last is sustainable and green processes. Enzyme-based biofuel production offers environmental benefits compared to traditional fossil fuel-based processes, but sustainability concerns remain. Future developments will focus on improving the sustainability of enzyme production processes, including the use of renewable feedstocks, energy-efficient fermentation methods, and waste minimization strategies. In conclusion, enzymes have the potential to revolutionize biofuel production, paving the way for a more sustainable and secure energy future. By addressing the existing challenges and continuing research efforts, we can unlock the full potential of these biological catalysts.